welcome to all of you. Today we are here to discuss DNA-protein interaction with special reference to nucleosome model. The genetic material of all organisms and viruses takes the form of tightly packed nucleoprotein. It exists as a compact mass that is confined to a limited volume, and its various activities such as replication and transcription must be accomplished within this space. The organization of this material must accommodate transitions between inactive and active states. In any living system, the length of the DNA as an extended molecule greatly exceeds the dimensions of the compartment that contains it. The DNA must therefore be compressed very tightly to fit into the space available. This remarkable feat of compression is performed by proteins that coil and fold the DNA into higher and higher levels of organization. The degree to which DNA is condensed is expressed as its packaging ratio, which is the length of the DNA divided by the length into which it is packaged. This packaging ratio in eukaryotes reaches 1000 to 2000 in interphase nuclei and up to 10,000 in metaphase nuclei. For example, the smallest human chromosome contains 4.6 into 10⁻⁷ base pairs of DNA, about 10 times the genome size of the bacterium E. coli. This is equivalent to 14,000 micrometer or 1.4 centimeter of extended DNA. At the most condensed moment of mitosis, the chromosome is about 2 micrometer long. Thus, the packaging ratio of DNA in the chromosome can be as great as 7,000. The DNA of interphase chromosomes, although less condensed than that of mitotic chromosomes, is packed tightly with a compaction ratio of about 1000-fold. Not only do chromosomes condense and relax in concert with the cell cycle, but different regions of the interphase chromosome must unpack to allow cells to access specific DNA sequences for replication, repair, or gene expression. Chromosome packaging must therefore be flexible enough to allow rapid, localized, and on-demand access to DNA. To achieve the overall packing ratio, DNA is not packaged directly into final structure of chromatin. Instead, it contains several hierarchies of organization. The first level of packaging is achieved by the winding of DNA around a protein core to produce a bead-like structure called a nucleosome, giving a packaging ratio of about 6 and produces 100 angstrom diameter chromatin fiber. The second level of packaging is the coiling of beads in a helical structure called the 30 nm fiber that is found in both interphase chromatin and mitotic chromosomes. This structure increases the packaging ratio to about 40. The final level of packaging is determined by the organization of 30 nm fiber itself. This gives an overall packaging ratio of about 1000 in euchromatin, cyclically interchangeable with packaging into mitotic chromosomes to achieve an overall ratio of about 10,000. Heterochromatin generally has a packaging ratio of about 10,000 in both interphase and mitosis. Several models have been proposed to explain the association of DNA and protein in the chromosomes. These models have been grouped under two heads: multi-strand models and single-strand models. In the multi-strand models, the chromosome is supposed to consist of several DNA protein strands. Each strand or fibril is supposed to consist of 20 to 40 angstrom DNA double helix associated with proteins. The single-strand models claim the existence of a single DNA molecule which runs along the entire length of the chromosome from telomere to telomere. They are uninterrupted through the centromeric region. Thus, the chromosome structure has been demonstrated to represent a unanemic skeleton. Regarding the manner of coiling and folding of DNA, a number of models are available, but only two of them stand out and are important. These are folded fiber model and nucleosome model. The folded fiber model was proposed by Dupra in 1965. According to this model, the bulk of the chromosome is visualized to be composed of a tightly folded fiber which has a rather homogeneous diameter of 200 to 300 angstrom. This folded fiber is supposed to contain the DNA histone helix of 30 angstrom diameter in a supercoiled condition. The nucleosome model was proposed by Roger D. Kornberg in 1974 and confirmed by Audit and his co-workers in 1975. This is the most significant and universally accepted model. In the nucleosome model, it was proposed that the histones were bound on the inside of the DNA coils, that is, histone particle with DNA around it. From a genetic perspective, a significant feature of packaging mechanism through the nucleosomes lies in its topology. That is, at no point is the DNA buried. Instead, it is freely exposed along the entire surface of the spool, available for genetic expression. Nucleosome seems to be a universal device for compacting the long DNA molecules of eukaryotic cells. Now, the structure of the nucleosome. When interphase nuclei are suspended in a solution of low ionic strength, they swivel and rupture to release fibers of chromatin. In some regions, the fibers consist of tightly packed material, but in regions that have become stretched, they can be seen to consist of discrete particles called the nucleosomes. Individual nucleosomes can be obtained by treating chromatin with the micrococcal endonuclease, which cuts the DNA thread at the junction between nucleosomes. First, it releases groups of particles and then it releases single nucleosomes. The nucleosome contains about 200 base pairs of DNA wrapped in a left-handed superhelix around histone octamer that consists of two copies each of the histone molecules, that is H2A, H2B, H3 and H4. These are known as the core histones. The core histones are present in equimolar amounts with two molecules of each per 200 base pairs of DNA. The H3-H4 tetramer occupies a central position in the octameric core structure, flanked on both sides by the H2A, H2B dimers. The octamers are assembled by the successive addition of two H2A, H2B dimers to the H3-H4 kernel. The shape of the nucleosome corresponds to a flat disc or cylinder 11 nanometer in diameter. The height of the cylinder is 6 nanometer, of which 4 nanometer is occupied by the two turns of the DNA. The pattern of the two turns has a possible functional consequence. One turn around the nucleosome takes about 80 base pairs of DNA. So, 2 pointers separated by 80 base pairs in the free double helix may actually be close on the nucleosome surface. The length of the DNA is roughly twice the 34 nanometer circumference of the particle. DNA follows a symmetrical path around the octamer. Path of DNA around the histone octamer creates about 1.65 supercoils. The DNA enters and leaves the nucleosomes in the same vicinity at points close to one another and could be sealed by histone H1. The association of DNA with the histone octamer forms a core particle containing 146 base pairs of DNA. They are irrespective of the total length of DNA in the nucleosome. This complex of the histone octamer and 146 base pairs of DNA is known as the nucleosome core particle. Nucleosome, that is nucleosome's core particle, linker DNA and linker histone, is a fundamental unit of transcriptionally inactive chromatin in all eukaryotic chromosomes, and most of its sequence is in the inactive state for most of time. The variation in total length of DNA per nucleosome is superimposed on this basic core structure. This suggests that the nucleosomal DNA can be divided into two categories: core DNA and the linker DNA. The core DNA has an invariant length of 146 base pairs and is relatively resistant to digestion by nucleases. Linker DNA, that is the DNA between core particles connecting nucleosomes and chromatin, comprises the rest of the repeating unit with a length varying from as little as 8 base pairs to as much as 114 base pairs per nucleosomes. All the 4 histones that make up the nucleosome core are relatively small proteins with a high proportion of positively charged amino acids, that is lysine and arginine. The positive charges help the histone bind tightly to the negatively charged sugar phosphate backbone of DNA. These numerous interactions explain in part why DNA of virtually any sequence can bind to a histone core. Each of the core histones also has a long N-terminal amino acid tail which extends out from the DNA histone core. These histone tails are subject to several types of covalent modification that control many aspects of chromatin structure. Histone H1 comprises a set of closely related proteins that show appreciable variation between tissues and between species. The role of H1 is different from that of the core histones and is present in half the amount of the core histone. It can be extracted more readily from the chromatin. The histone H1 can be removed without affecting the structure of the nucleosomes, which suggests that its location is external to the particle. The nucleosomes are further packed upon one another to generate a more compact structure, the 30 nm fiber. This appears to be a solenoid structure with about 6 nucleosomes per turn and a packaging ratio of 40. Removal of H1 allows this fiber to unfold into a 10 nm fiber that consists of a linear string of nucleosomes. The 30 nm fiber probably consists of the 10 nm fiber wound into a two-start solenoid. Formation of solenoid from nucleosomes can be compared with winding of a cable, that is DNA, on a spool, that is nucleosome, and then folding of wrapped spools. Stability of this structure requires the presence of the last member of the histone gene family, that is histone H1. The 30 nm fiber is the basic constituent of both euchromatin and heterochromatin. 30 nm solenoid fiber is organized into looped domain structure. Each loop has approximately 50 turns of solenoid. These loops are thought to undergo at least one more level of packing to form the mitotic chromosomes. This final level of packaging is seen in the metaphase chromosome. The condensed piece of chromatin has a characteristic scaffolding structure that can be detected in metaphase chromosomes. The scaffold is made of non-histone proteins and retains the general shape of the metaphase chromosome. The scaffold proteins include two abundant proteins, the larger DNA topoisomerase II and the smaller matrix protein. Both initiation and continued replication of DNA occurs in association with matrix proteins and topoisomerase II binding sites. The binding sites for topoisomerase II are called scaffold attachment regions, and matrix proteins bind matrix attachment regions. It is believed that the same sequence of DNA works as matrix attachment region in interphase nuclei and as scaffold attachment region in metaphase chromosomes. Each chromosome has two scaffolds, one for each chromatid, and they are connected together at the centromere region. When the histones are removed, the DNA, which has packed about 40-fold in the 30 nm chromatin fiber, becomes extended and produces loops with an average length of 25 micrometer. In each loop, the DNA exists from the scaffold and returns to an adjacent point. On the basis of these observations, a model of chromosome structure called radial loop model has been proposed by Lamilly and his co-workers in 1984. According to this model, DNA is arranged in loops anchored to the non-histone scaffold. Because the later loops have 25 micrometer DNA, and after contracting 40-fold in the 30 nm fiber, they would be only about 0.6 micrometer long, a length consistent with the diameter of metaphase chromosome, that is 1 micrometer. The chromatin in mitotic chromosomes is transcriptionally inert. All RNA synthesis ceases as the chromosomes condense. Probably, the condensation prevents RNA polymerase from gaining access to the DNA, although other controlling factors might also be involved. Packaging promoters in nucleosomes prevents the initiation of transcription by bacterial and eukaryotic RNA polymerases. Both histone fold DNA interactions in the core particle and histone tail interactions in the chromatin fiber are thought to contribute to repression, and each is counteracted by specific mechanisms. Repression due to interactions in the core particle is opposed by chromatin remodeling complexes, while that due to condensation in the chromatin fiber may be relieved by histone style transferases and re-established by histone deacetylase complexes. A number of observations suggest that de-repression at the level of the chromatin fiber precedes that at the level of the individual nucleosome. Now, the modification of nucleosomes. Although the nucleosome is a very stable protein DNA complex, it is not static and has been shown to undergo a number of different structural rearrangements, including nucleosome sliding and DNA site exposure. This packaging can be dynamic, allowing rapid access to the underlying DNA, which carries enormous amounts of coded information, and it is especially important that cells have on-demand access to it. The direct access to DNA is typically localized, exposing only those regions of the genome that the cell needs at any particular time. Eukaryotic cells have several ways to rapidly adjust the local structure of their chromatin. Nucleosome structure is altered by nucleosome remodeling factors, facilitating the binding of transcription factors to chromatin by repositioning nucleosomes on the DNA through sliding of histone octamers along the DNA molecule. Chromatin remodeling complexes are the protein machines that use the energy of ATP hydrolysis to change the structure of nucleosomes. These complexes can make the underlying DNA more accessible to other proteins in the cell, especially those involved in DNA replication, repair, and gene expression. During mitosis, at least some of the chromatin remodeling complexes is inactivated, which may help mitotic chromosomes maintain their tightly packed structure. Nucleosome undergoes histone modifications to become transcriptionally active. The core histones share a structurally conserved motif called the histone fold and mobile extended regions at the chain termini, also named histone tails. The histone fold consists of 3 helices alpha 1, alpha 2, and alpha 3, connected by short loops L1 and L2 respectively. During dimerization, loop L1 of one of the monomers, for example H3, aligns against the loop 2 of the other monomer, that is H4, to form the so-called handshake motif that interacts with DNA. The flexible tails of the core histones interact with DNA via the minor groove. The histone tails are the major targets for post-translational modifications such as acetylation, methylation, ubiquitination, and phosphorylation, and so are the key arbiters of chromatin function. The amino-terminal tail domain of histone undergoes the modifications to increase the accessibility of chromatin to DNA-binding proteins for transcriptional activation. Combination of specific histone modifications constitute a histone code that regulate gene expression by recruiting other regulatory proteins to the chromatin template. As is true for the chromatin remodeling complexes, the enzymes that modify histone tails are tightly regulated. They are brought to a particular region of chromatin by other cues, particularly by their interactions with proteins that bind to specific sequences in DNA. It is likely that these histone-modifying enzymes work in concert with the chromatin remodeling complexes to condense and relax stretches of chromatin, allowing local chromatin structure to rapidly change according to the needs of the cell. The coupling between the histone octamer and the surrounding DNA superhelix is instrumental in regulating the dynamics of the octamer, and conversely, the closely interacting proteins significantly alter the dynamics of the DNA, signaling the important role of the complexation with histones in regulating the transcriptional activity. Now, the significance of nucleosomes. The nucleosome is fundamental to DNA coiling and gene regulation. The nucleosomes efficiently package DNA and create an added layer of regulatory control, which ensures correct gene expression. Nucleosomes are thought to carry epigenetically inherited information in the form of covalent modifications of their core histones. The maintenance of a repressed or activated gene is often necessary for cellular differentiation. Dear students, we have discussed over here packaging of DNA and the various models of packaging of DNA. Dear students, we have also discussed here structure, modification, and the significance of nucleosomes. Thank you very much. God bless you.